till where we have done in the last class? Yes, codominance over one gene, two gene, incomplete codominance. All the four over. Yes, chromosomal theory of inheritance. No. After studying about the one gene inheritance, two gene inheritance, incomplete dominance, and codominance, four different phenomena. So, two scientists by name Sutton and Bovary. So, they proposed a chromosomal theory of inheritance. So, what exactly is this chromosomal theory of inheritance? It's the by taking the concept of Mendelian principles and by studying the movement of chromosomes during meiosis. So, by taking the Mendelian principles, law of dominance, law of purity of gametes, law of independent assortment, and at the same time studying the movements of chromosome during meiosis. So he joined both together, both the concept he took together, and the both the person Sutton and Bovary proposed the theory called chromosomal theory of inheritance. What Sutton and Bovary wanted to tell us is whatever Mendel has told in the concept. Right? Number one, law of dominance. What Mendel told in the law of dominance, when a pair of genes are present together, one will be dominant, one will be recessive. Yes, he proved the law of dominance. Then second, law of purity of gametes. He told us about the purity, how the gametes were present together. Later, they segregated apart. Blending did not took place. And third, independent assortment, how they occupied the particular position in the Punnett square. Yes. So whatever has happened in the genes, no. So first it will happen in chromosome. According to Sutton and Bovary, chromosomal theory of inheritance, what they are proving, they wanted to tell us that whatever has happened in the gene, that will happen first in the chromosome. Why? Why? Yes, genes are located on the chromosome. Is that clear? So, according to Sutton and Bovary, if you see the genes are occurring in pair, yes, the genes are occurring in pair, yes. At the same time, chromosome also occurs in pair. Yes or no? So, segregation of gametes takes place. No? The segregation of gametes. So chromosome also segregates. Yes. Assortment will take place. Assortment of genes take place. Similarly, assortment of chromosome. Arrangement of chromosome. Chromosome occupying particular position. So whatever happens in the genes, first it will happen in the chromosome. Is that clear? It will happen in the chromosome. So according to the chromosomal theory of inheritance, the chromosomes are responsible for the dominance, for the segregation, for the assortment. Is that clear? When the chromosome does not occur in pair, gene will occur in pair? No. If the chromosome does not segregate, gene will segregate? No. If the chromosome does not occupy a particular position after assortment, the gene will occupy? No. So whatever Mendel has proved, Sutton and Bovary wanted to prove that the according to chromosomal theory of inheritance, whatever the inheritance of character that is happening from one generation to another, no, that is because of chromosomes. Is that clear? Did you understand? Now, so they this theory was discovered by Sutton and Bovary. It was discovered by Sutton and Bovary. But this theory was experimentally proven. It was experimentally proven by whom? Yes, T.H. Morgan, that is Thomas Hunt Morgan. Yes. So, to prove this chromosomal theory of inheritance, Thomas Hunt Morgan, what he did, he took the organism. Which organism? Drosophila melanogaster, which is also called the Cinderella of genetics. Yes, it is also called as Cinderella of genetics. Did you understand till here? Yes. Now, question arises: why did T.H. Morgan took the Drosophila melanogaster as the experimental species? What was the reason? Mendel took pea plant. There was the reason behind. No? To conduct experiment, he selected pea plant as the experimental plant. Simultaneously here, T.H. Morgan chose? Yes. Why? What are the features that he was able to see? Features are characters because of which he took Drosophila melanogaster. No? Yes. Number one, it can be easily grown on synthetic medium. Yes, majority maximum sources are not required for the growth. Any one or two growth source available in a particular place, the organism starts growing and multiplying. Is that clear? Then number two, yes, short lifespan. No? It will complete the whole life cycle in 2 weeks. Short lifespan. Yes. Yes. Single mutation can lead to many variations in progeny. Even a single mutation can lead to many differences, many variations in the offsprings. Yes. Male and female are separate. easily male and female can be identified. They are separately present. Anything else? Yes, yes. Close your book. नहीं फीचर की स्टोरी मालूम रहे तो देखने का मजा नहीं नहीं 3 घंटे वेस्ट करे सर का नहीं यस नाउ यू टेल limited number of chromosomes. So limited number of chromosome. How many chromosomes? Yes, yes. N equal to 4. 2n equal to 8. Done. So 2 mark question in theory. In the theory, very commonly asked 2 mark question. Why Drosophila melanogaster is used as the experimental species by T.H. Morgan? Yes, these are the few reasons. It can be easily grown on the synthetic medium, short lifespan. In 2 weeks, it will complete its whole lifespan and it will die. Then, single mutation can lead to large number of variations. Many variations can be seen. Male and female are separate, easily identified, and the number of chromosomes are also limited. 2n equal to 8, n equal to 4. This was the reason. Yes. Any doubts still here? Yes. Next, after studying about the chromosomal theory of inheritance, no, we need to study two very important small topics. One is called as linkage and recombination. This is a connection to chromosomal theory of inheritance. When we study linkage and recombination, that's a connection to again chromosomal theory of inheritance only. Yes. So here, number one, what linkage? Any idea what is this linkage? Yes, linkage. No, no idea. You have not studied? Huh? Studied, but you don't remember? No, sir. Not yet. We have studied. We have not listened. Eh? Tell me. Tell. You don't remember, or you have not studied yet? पढ़े नहीं पढ़ को ही नहीं पढ़ को ही हाँ भूल गए हाँ yes so very important topics from which we can study the chromosomal theory of inheritance. Is that clear? What is this linkage? So here physical association of genes on a chromosome. So here physical association of genes. The question is which genes? No? So here physical association of parental genes. Parental gene. So here usually assume this is the chromosome A, B, A, B. What type of chromosome? This is a homologous chromosome. Homologous chromosome, identical. Yes. So in this, if the genes are present, a pair of genes are present. For example, gene number 1 is present here, gene number 2 is present here. Is that clear? And identical. Again, gene number 1, gene number 2 is present here. Ne? Now, these genes, when they are transferred from parental generation to the next generation, offspring, when they are present in the parental generation and easily they are transferred to dominant, they are also expressing. Parent is also expressing this gene, offspring is also expressing the gene. Now again, if this gene is again transferring into the next generation, second generation, third generation, means the gene is associated from the parent. Assume first grandfather, father, Now son, grandson. So two generation, three generation, four generation, continuously the link is there. There is no stoppage. Is that clear? Connection is continued. So such genes which are transferred from one generation to another, and when they are expressing themselves, no, this phenomena is called as linkage. Physical association. Physical association of parental gene on the chromosome, through which the particular character is expressed in all the generations. Is that clear? So why this is possible? So if you see, two genes are present, two pair of genes are present. Now if the next generation is appearing, again these are getting transferred easily. Yes, it will be transferred again here. In the next generation we are also we are able to see. Yes, then again third generation. Why? Because linkage, when it will happen, when the genes are nearby, when they are present on the homologous chromosome, and the closer genes which are present together, no, easily they get transferred. So that frames linkage. Is that clear? Assume similarly I will take a homologous chromosome. Yes, but the genes which are present are far. They are far. They are far. Now this connection is easy, or this connection is easy? This one? Yes. So the genes which are closer, and when they are transferring from one generation to another, that association, that phenomena is called as linkage. Here also it possibility is there. The linkage will take place, but complete linkage will not take place. It will not take place completely. So to this process, to this phenomena, we can call it as incomplete linkage, incomplete linkage, or we can also call it as recombination. What happens in the recombination? Instead of physical association, no, so here the physical association is taking place between non-parental gene combination. Is that clear? When the parental genes are getting transferred continuously, it is linkage. Non-parental, when non-parental association joining, here number one is there, combination is taking place like here it is happening. Again, if you take, it will be non-parental. One is not matching with one, two is not matching with two, no, so non-parental. One matching with one, two matching with two. Continuously from generation to generation, the same genes are getting transferred. That is linkage. There, combination will take place, but in different manner. So here, when the genes are far, when they are not combining, is it clear? So in the one generation you are able to see, in second generation you are not able to see, third generation again it is combined, no? Particularly any character which you seen in the grandfather, in father it is not present, again in the grandson it is come. That means it is recombination of genes. Did you understand? Yes. So linkage and recombination, two very important one mark questions. What is linkage or what is recombination? Then physical association of parental genes on the chromosome is linkage. Non-physical association, non-parental gene combination. Is that clear? Physically also it is not combining. So recombination has to take place. Such phenomena is called as recombination. Done? Yes. Yes. Next, we have done with polygenic inheritance and pleiotropy. No? Yes. Yes. Polygenic, two very important one mark questions. You have written test now. Board test. One question was asked related to multiple choice. No. Yes. Pleiotrophy. Yes, pleiotrophy and polygenic inheritance. Two very important multiple choice questions. This question is very common. Either they will ask you related to polygenic inheritance or pleiotropy, or one more. Next, I will go mutation. From these three topics, any one multiple choice question or fill in the blanks will be continuously, repeatedly asked. Yes. Now, polygenic inheritance. What is this polygenic inheritance? Yes. Expression of trait controlled by two or three or many genes. No, one gene inheritance we studied. Nay, two gene inheritance we studied. Now, if it is more than two, it is polygenic inheritance. So here, easy example. We can take example of height, or we can take example of skin color. Yes. So expression of trait. Height is one trait or character. Skin color is one trait or character. No. So now if I tell height, in one gene inheritance what we studied, height means tall, tall, dwarf. Yes, only two genes. Yes, only one gene. Height, one character we took as tall, dwarf for height. Right? Now only two genotype will be seen. Is there any other genotype? If you take height, only we are able to see tall and only short. Medium nahi hai. Medium size hai nahi. Yes. Intermediate. Huh? If you take skin color, yes, if you take skin color, no, fair, yes, white, black, nahi. How many you are able to see? Three. Nahi. Any. Assume. If you take tall and draft, nahi. So for this, if you take tall, again for tall also, they will be dominant character and recessive character. Nahi. So here, if you take capital A, capital B, capital B, capital C, capital C. Yes, so we have taken the dominant form. Nay, for this, if we take recessive form, small a, small a, small b, small b, small c, small c. Now, if you cross this two, yes, what will be the result? Capital A, small a, capital B, small b, capital C. Yes. So, how many pair of genes are controlling the character? Three. Yes. So, this phenomena is called polygenic inheritance. Is that clear? Now, to this same opposite, expression of trait controlled by two or more gene. Now, here, one gene expression, one gene is controlled by many phenotypic expressions. A single gene is controlled by opposite. Here one trait is controlled by many genes. Here one gene will be controlled by many phenotypic expressions. Is that clear? Easy example we can take phenyl ketonuria. Yes, you've studied. You've studied this disease, one of the genetic disease, Mendelian disease. What exactly happens in this? Yes. Why we are using this disease as an example for pleiotropy? No idea? Yes. One gene controlled by many phenotypic expressions. Ne? So here, when we talk about phenylketonuria, this is one of the genetic disorder that is seen in the humans. What exactly happens in this? Phenylketonuria. Yes. So this disease is autosomal recessive disease found in the individual where the person will be suffering from inborn error metabolism. The person will be suffering from inborn error metabolism. So here in our body 24 bar 7 the metabolic activity is taking place. Eh? What is metabolism? Yes, sum total of catabolism. Huh? Yes, the sum total of all the reactions. Anabolism as well as catabolism. Only catabolism is taking place. Anabolism also. What is anabolism? Then what is catabolism? Huh? We should learn, no? फर्स्ट ईयर में पढ़े नहीं क्या जी? फर्स्ट ईयर में फर्स्ट चैप्टर पढ़े नहीं, लिविंग वर्ल्ड? हाँ, कल पढ़ा सोच याद नहीं. Yes. Catabolism is the breakdown process. Whatever is happening no in our body, when the process is breaking, नहीं? You are eating food. Food is getting broken down into micromolecules. नहीं, macro से micro होता है. That is catabolism. ठीक है? After breaking, it is going to your stomach, then small intestine. From that, amino acids are observed. From that, proteins are getting build up. No, that which is getting build up is anabolism. Anabolism is build up process. Catabolism is breakdown process. Together it is metabolism. No, so here what exactly happens in this inborn? Why we are telling inborn? By birth the error will take place. Why? Because one gene is absent in the person. Only one gene is absent in the person, or only one enzyme is absent in the person. So here usually when we consume food, when we eat food, no, so usually the phenylalanine, no, this phenylalanine has to be broken down. It has to be broken down. So when it is broken down, this will be converted into tyrosine. In our body it has to be broken down and it has to be converted in the form of tyrosine. Yes, in the normal individual this is happening, but if the person is affected, if that one particular enzyme, which one? Phenyl hydroxylase enzyme, this enzyme is present in us, so we are normal. We are normal. Is that clear? So if the person is not having this particular enzyme which will convert phenylalanine into tyrosine, so usually what happens, the if it is absent, instead of tyrosine, this phenylalanine will get converted into phenyl pyruvic acid. Phenyl pyruvic acid. Yes, what we are studying, we are studying about pleiotropy. Nay, to study, to understand pleiotropy, we have taken the example of phenyl ketonuria. Yes, Now, what I told in the pleiotropy, definition of pleiotropy, single gene controlled by many phenotypic expression. Because of this one gene absent in the person will show many symptoms. Many symptoms. Number one, the person will be mentally retarded. The person will be mentally retarded. Why? What will happen? Kidneys will not function properly. Kidney functioning is disturbed. Once the person is lacking this one gene, the metabolic activity is disturbed. Metabolic activity is disturbed. Because of that chemical reaction not taking properly, the lower performance of the kidney. No? So here, the pyruvic acid which is produced, the pyruvic acid which is formed in the organism body, it start accumulating in the whole body. If tyrosine is produced, that will be taken up by the liver. Actually, it's a liver enzyme. Okay? It will be taken up by the liver. Then it will send to the kidneys. Filtration will be done. We are normal. Ne? If the enzyme is absent, conversion has taken into another part. Instead of tyrosine, pyruvic acid is formed. Yes, that pyruvic acid is not further broken down by your kidneys. So usually what happens? The whole body, kidneys also, will be accumulated by the pyruvic acid. Finally, after your whole body gets accumulated, your kidneys get accumulated, finally it will reach your brain, and your brain also will be accumulated by pyruvic acid. Therefore, the person will be mentally retarded. Is that clear? So here your brain is retarded, your kidneys failure, and other symptoms what the person is showing. No, because of that, the person is suffering from the disease. So many phenotypic expression because of one gene controlled by many phenotypic expressions. Did you understand? Yes. What is linkage we know? What is recombination we know? What is polygenic inheritance we know? What is pleiotropy we know? Done? Any doubts? No doubts? Yes. Next. Yes. This is also in this chapter it is one mark question. In this chapter it is only one mark question will be asked. In the next chapter it is a 2 or 3 mark question in the molecular basis of inheritance chapter. Yes. What is this mutation? Have you ever heard this word mutation? Yes. Sudden change. Yes. Sudden change. Yes. Only sudden change where? Yes. In the genes, in the DNA, sudden change. Yes. We can tell addition or deletion of genes or alteration of genes in a cell. So addition or deletion or some alteration happens. So when this happens, no, single, usually a single base pair change will take place, or two genes will shift their position, or addition or deletion will take place, or finally a codon also can be altered. So this can lead to a single step change in the processes that are taking place in a cell. What are the basic processes that are taking place in our cell? Yes, we are eating food. Yes, we are eating food. After eating food, digestion process is taking place. What is the result of digestion process? Why are you eating food? To get energy in the form of, yes, proteins. Yes, directly you will get proteins. No, what we are taking is amino acids. Nay, whatever we are eating food, from that food, nay, the carbohydrates which we are taking, so that carbohydrates will give us amino acids, and that amino acids will be taken up by our body, by our cells, and in the cells, no, inside the cell, inside the nucleus, inside the chromosome, inside the chromatin fiber, inside the nucleosome, where in the DNA, no, so two process will take place. Two process will take place. One is called as transcription, and translation. Yes. So basically, the mutation in our body, occurs during transcription process and translation process. Yes. So what will happen, sir? If mutation takes place in our body during this process, what is transcription, what is translation, in the next chapter you will study in detail. In short, I will tell you, transcription is you are copying the information. Two strands of DNA are present. Yes. The information is present here. Yes. The information is present here. Yes. From one strand of the DNA, you are copying the information to RNA. You are copying that to mRNA. Is that clear? This process where the information is copied from the parental strand of the DNA to mRNA is called transcription. Is that clear? Why this information is required? To undergo with the translation process. What is translation process? In short, it is protein synthesis. The amino acids which you are having, no, after eating food, when you have taken the amino acid, that has to get converted into proteins. To convert that into proteins, your DNA is involved. Is that clear? Your RNAs are involved. Three different RNAs are involved. Eh? mRNA, tRNA, rRNA. Three are involved. With the support of DNA and RNA, the amino acid forms a long polypeptide chain. That is nothing but protein. Is that clear? So translation. So during this transcription and translation process, mutation occurs. Mutation occurs. What exactly happens, sir? So here, in short, simple words, in the next chapter also same we will study. In simple words, I will tell you. So here, for example, Ram has big cap. Yes. If you see, I have taken three letters. No. So here, why? Because the in the DNA, if you see the nucleotide bases which are present, the nucleotide bases which are present, they will be present in the form of codon A T G C, A T G C, like this. Three they are present. Yes. So this is called as a codon. It is all called as codon. Yes. So mutation, when it occurs in the DNA, no, continuously, if you see the codons are present in the DNA. Assume this is one DNA strand. Yes, the codon is present in the DNA strand. Now, if mutation occurs, if the mutation occurs only in one base, single nucleotide base is erupted. Right? For example, remove B first. If you remove B, no, Ram has IGCAP. Yes. Again, the strand is having continuously. Yes. You are able to see it has become discontinuous now. Yes. Only one base pair. Only one base pair is removed. It is removed. So the whole DNA strand has been disturbed. This is only called as mutation. And when one base pair is removed, the mutation is called as, it is called point mutation or gene mutation. If one base pair is removed, it is called point mutation or gene mutation. Okay. Now assume I will remove I also. Now Ram has GCAP. Yes. So two genes are removed. Yes. Now it is called as chromosomal mutation. It is called, if one base pair is removed, it is called point mutation. If two are removed, it is called chromosomal mutation. If three are removed, I will remove G also now. Ram has, yes, C A T. Done. It became continuous again. Yes. Now this when we are removing a complete codon, it is called frame shift mutation. Yes. What is mutation? You know now. What are the three different types of mutation? Yes. Coming to the example, if you take example of point mutation, yes, point mutation, sickle cell anemia, no, sickle cell anemia. What exactly happens in sickle cell anemia disease? Yes, what I should understand? Huh? The red blood cells are concave shape. Yes, the shape of the red blood cell changes. It will become sickle shape. Why? Because the hemoglobin in the blood is controlled by a particular gene, HbA gene, and other HbS gene is controlling. Defective genes controlled by HbS. Good genes controlled by HbA. Coding is taking place. If you see, the coding is taking place. No? So, if, for example, CTC, CAC. So here G A G G U G. So this is controlled by HbA gene, and this is controlled by HbB, or in few books it is given HbS gene. The hemoglobin production which is taking place, good hemoglobin production is controlled by HbA gene, and the bad defective hemoglobin is controlled by HbS gene. So usually what happens, no, continuously, as I told you, I showed here. Nahin? So here codons are present. Codons are present. That codons are coding continuously amino acids. One codon will code for one amino acid. Nahin? So valine, proline, leucine, threonine, histamine, glutamic acid. So like this, in the normal, hemoglobin production, okay, in the blood, when the normal hemoglobin production is taking place, the positions occupied by the amino acid, first valine will be coded, in the second position the leucine will be coded, like this, the first position, second, third, fourth, fifth, sixth, seventh, like this, the positions will take place. Is that clear? So here in the sixth position glutamic acid is present. during the normal hemoglobin production. In the sixth position, glutamic acid should be produced. But what happens because of mutation of single base pair, instead of HBA, now HBS will code. Is that clear? Single shift, that is CTC, CAC. Instead of T, A is coded. So when T is coded, it will be coded by Y, because complete strand of DNA. Here in the RNA, T is not present, thymine is not present, uracil is present. So uracil will code. So because of this one base pair coding, no, here the amino acid coding will be changed. In the first position itself, valine is coded. No, but what happens again in the sixth position, valine will be coded. Defect. Okay. So glutamic acid have to move to seventh position. Is that clear? So this one change, only shift in the position. Gene is not absent here. Gene is present. Only shift in the position. Single gene is shifted the position. I told you addition of gene, deletion of gene, or alteration of genes. One alteration. No. So here what happens? So because of this, the oxygen production will be reduced. Supply of oxygen will be reduced to the red blood cell. So that red blood cell, when a proper level of oxygen is not supplied to the red blood cell, it will change its shape. From normal shape, it will get converted to sickle shape. Then once it becomes sickle shape, it start destroying itself. Sickle cell anemia. What is anemia? What is anemia? Sickle cell, now you got to know. Sickle cell disease name is sickle cell anemia. No, sickle cell you got to know that the normal red blood cell from the concave shape, it got converted to sickle shape. Nay, from the normal shape, it got converted to moon shape, sickle shape. Yes. What is anemia? Meaning of anemia: destruction of red blood cells. The red blood cells start destructing itself. Is it clear? The number of red blood cells become decreased. Did you understand? So here this is happening because of only one gene. No, we will tell gene mutation or point mutation, sickle cell anemia. Then chromosomal, second, two I told. Nay, chromosomal mutation, two nucleotides are changed. Nay, so chromosomal, what happens in chromosomal? Chromosomal disorders you have studied? Yes? If there is any defect in the genes, we will tell genetic disorder. Nay, Mendelian disorder. If there is any defect in the chromosome, chromosomal disorder. Nay. So here in your